Well, there's some cool pictures here. That's why I want. And you've got them on your iPads and laptops and whatever. Hopefully you're not looking at this on your phone, but whatever works. Okay. So let's talk neuroscience and what this means. So fundamentally, our approach still is very much centered on the nerve cell. Yes, we know a lot about nerve circuits. We know a bit about parts of the brain talking to other parts of the brain. Huge amount of work over the last decades, more than 50 years, looking at how one area of the brain talks to the other part of the brain. But how circuits are activated and what that means is still not fully understood. For example, why do we have emotions? Well, physiologically and in terms of adaptive biology, we understand we have emotions. Emotions actually color all of your memories, and they basically set them in almost a priority range. Now, if you have traumatic memories, that's a different story. That goes into a whole different category. So it's almost, the pathology of the emotions motive system, but nonetheless, emotions basically color everything we do. And so that can have a big impact on the recall, the accuracy, and so forth. So we know we have that, we know that it's important, it has a function, but how do they happen? We don't know. We don't understand most psychiatric disease. We can talk about neurochemical imbalances and receptors changing and all that and give drugs, but a lot of it's slipping at that level, but at a higher level we don't understand it. So a lot of what we do in neurobiology still revolves around understanding nerve cells. Now there is some question, really even to this day, about how in the human brain, how many different types of nerve cells are there? Now we know there's about 50 billion nerve cells, but how many types are there? And we see certain characteristics. We see often a cell like this. So this cell was produced by this individual, Santiago Ramon y Cajal, who's been often named the grandfather of neuroscience, which is a little bit, maybe the godfather. A little bit of an overstatement, but he was an extraordinary scientist working almost in obscurity in Spain for many years, producing these amazing images. And the thing that's amazing about this is, I'm not sure that's amazing, that's interesting about this, is that this staining technique, so this is a section of a brain, okay, the cortex actually of a mouse brain. And what he had done is he took a cocktail of chemicals to get the stain. But what he did is he banked it off of a stain by Camillo Golgi. So Golgi was truly the great neuroanatomist of the late 19th century. He's an Italian neuroanatomist. And a lot of things are named after him, right? Like the Golgi apparatus, the Golgi tendon organ. And those are only a few. There are not quite a dozen things that Golgi put his name on. So he's a tremendous anatomist, not just neuroanatomist. And he invented this stain. And what he found was that if he basically oxidized a tissue, he fixed it and oxidized it, and then he added in a silver compound, the silver would stain some of the cells, not even 5% of the cells in that tissue, but it would stain them completely. Well, actually not. Come back to this. Well, this guy comes along, and what's intriguing about Cajal was that he started out in art. He loved to draw. He sketched and he did watercolors, but he really loved these images, and he spent 50 years of his life creating these images and then drawing them. He was so exquisitely fine at doing that. And better still, he learned about this technique actually from someone else. And he tried it, and it didn't work very well. So he did what was called double impregnation, and this is just classic. He took the stain, he stained the tissue, it wasn't very good, he washed it, and he stained it again. And all of a sudden, it lit up beautifully to the end. So when Golgi was using the stain, when he set it up, he never actually stained the cells all the way to their very ends. And because of that, this led to a complete battle between the two groups as to whether nerve cells have endings and whether they have synapses. Because Cajal came up with basically, a double dip, so he could see more of the cell. Came up with a technique where he could see the ends, and he said nerve cells end, they make synapses, and this fit with what Sherrington had shown. and he was awarded the Nobel Prize. But he was awarded the Nobel Prize at the same time as Golgi, and Golgi would never give it up. So he actually, in his Nobel Prize speech, he didn't acknowledge anything that Cajal did. He said, oh, the nervous system's all interconnected to each other, and there are no such things as synapses. So it was remarkable. But you can see the detail here. This is one cell. You can see little knobs on it, and you probably already know a little about dendrites and axons. Cell bodies, we'll come back to those little bits again, but I'm sure you're aware that the dendrites are the receiving end, and the axon or nerve fiber is the output, the communicating end. Okay. So the other thing was that even in thicker tissue, he was able to focus up and down, and basically he created overlays that gave rise to images like this. Okay. This is now in the cortex. These are called parietal cells because they have a pyramid-like shape. Okay. They have all these extensions here, the dendrites, and this slender, thin, smooth axon coming out of them. And so he drew these, and this is actually a laboratory in his kitchen, in his house. I don't know how his wife felt about that, but he worked all day, every day. He ate food, then he kept working, and he drew these amazing images. But he used something called a camera lucida. That was a way to project that down on a piece of paper and see all this extraordinary fine structure. And he was able to follow all these and look at their interactions, and he was saying, oh, some of these look like they're making these contacts. And often, that contact looked different than the body of the axon, and that, again, fit with the idea of the synapse. So, here's another image. So this one is in what we call the cortex of the brain. This actually might be this is, I believe, in the hippocampus, but it's in the cortex of this particular region of the brain, and you can see some kind of organization here. The cells, they're all unique in one manner of speaking, and yet you can see that they have a recognizable morphology, right? And then they seem to be vertical, right? So the information, if you had to say, was going that way, isn't it? And then there's these up here running longitudinally. And often what you'll see is that there'll be a layer where there's this vertical information, and it may just go down the next layer, or it may go out and go to a whole other part of the brain. Okay. Up here, these often connect different parts of the same region. And you see that kind of pattern over and over again. This, though, this is actually a section of the spinal cord. And the thing, I'm going to blow this up. So again, these are his drawings, but you could see that different types of fibers coming into what's called the back end or the dorsal part of the spinal cord. And so that's where the sensory information comes in. And you can see that they came to these little bulbous endings. We now call those boutons, from the French meaning button, but it's just a little nub in there. Golgi missed those. He would see something like that. They were all gone because he didn't stain it enough to get out there. All right. And then you could see some cases where there's little bifurcations. And then again, this is in the spinal cord, an enormous rich amount of fibers. And again, we're not staining even 5% with this technique. And this is something that neuroscientists, budding neuroscientists and even mature neuroscientists will look at and say, this is amazing. He was just doing neuroanatomy and just looking. So this is, again, from the cortex looking at organization, again, of a pyramidal cell. They're lumpy. They're not exact pyramids, okay? They're lumpy. Okay. Look at this, and he drew arrows. How the hell did he do that? First of all, there was really no information that this was truly the receiving end of the cell and that this would be the axon. Okay. There was some information from work functionally. But when you do functional work, you can't see it. Well, now we can, but in those days you couldn't see it. They drew arrows, and these arrows are correct. So one thing that was interesting, you could see a cell here that had an axon that then went over and made contacts with the neighboring receiving or dendritic part of this cell. Okay. The other thing is I think I have a blow up of this. This is actually not the best resolution. This one's better. This is from the original drawing. As a mark from the Cajal Institute in Madrid. But he could see that, particularly up there, he saw that these fibers went in contact with the cell body or the dendrites, all right? They never contacted anywhere on the axon, ever, ever, ever. Sometimes in certain unusual preparations, you can see little contacts near the nerve end, two nerve endings real close to each other. So that was a big clue as well. So from this, he developed the might be called the synapse hypothesis, that there are synapses. So now I want to stop for 2 seconds, maybe more than 2 seconds. And I'm going to turn the lights on because you can read this, right? So now, let's talk about synapses. So there's no question that the vast majority of contacts between nerve cells work between these synapses. Okay, and there's a gap, and we call it a synaptic cleft. The cleft is not the synapse is the whole thing. We'll come to that in a second. Okay, but it's a point of communication, there's a gap, and somehow we now know there's electrical activity on the axon that has to bridge from one cell to another. We're going to go into that because, as you know already, there's neurotransmitters, there's chemicals that do that. Not everywhere, there's such a thing as electrical synapse, but fundamentally in the mammalian brain, actually in vertebrates, the vast majority are, in fact, gaps or chemical synapses. Okay. So, first question. What simple common neurological test tells us that our nerves work through relays? What is a synapse? It's a relay, right, between two nerve cells. Reflex circuit. Did everybody hear that? So a neurologist can test virtually all of your muscles. Wherever a neurologist can get access to a ligament, a tendon, I should say. Let's be clear. Whether it's in your arm, your bicep, your triceps. Obviously, you know about the patellar reflex, right? You all tried to do that? You can go ahead and do that. Just don't kick the guy in front of you. But when you've done the patellar reflex or had it done to you, whether it was a doctor or somebody sneaked up on you and hit you on the knee, did it feel like it was instantaneous, or did it feel like you could tell there was a tap and then your leg went? Who wants to vote on that? Was it that your leg tapped and your knee went out, like it almost happened instantaneously, or did it feel like there was a little bit of a delay? Now don't say what you think. I want to know. You can say, yeah, it was instantaneous. What are you talking about? Most people would say, if I polled you, that it felt like there was a little bit of a delay. How short a delay do you think a human can perceive? I'll give you a minute. You can look it up if you want. Or you can guess. Actually, you can use something and help you guess. Who knows what video rate is? Come on, there's got to be a couple of geeks in the audience. Video rate. 33 frames per second. When you watch a video, does it flicker? So it was thought for a long time that we can't see that. So that's 33 milliseconds, right, between frames. Flash, 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 right? But it looks continuous, doesn't it? Well, you know, in computers, they don't refresh the whole screen. Okay, it doesn't matter. If you have a true video, full frames, 33 milliseconds. That should tell you that you can't see that, right? Ooh. So the feeling was that from early tests that we could see about 100 milliseconds. It would start flashing, right? Very recently, there was some tests done where a group would flash for less than 33 milliseconds, just colors, you know, pink, orange, yellow, green, whatever, and found that we can actually see an image if you just do it once at 13 milliseconds. So we can actually see better than video rate, but our brains, when we see video, want to blend it together. Some people actually can perceive video rate. You can test for that. But normally it doesn't seem like you can, right? It looks smooth, but it's probably jumping a little bit, but you just, you know, your mind kind of blends that in. You flash an image for 13 milliseconds, you can see it. Okay. So, any quicker than that, no. Now that's from your retina back to perception, right? There's a few synapses. But a reflex arc, all right, where you do patellar tendon or you touch a hot stove, right? How fast do you think that is? Let's start with your toe. Okay, so you stub your toe. This would be a pain pathway, not the best, but we're going to use the limits on it. Your pain pathway, you stub your toe, and what do you usually do? You actually have a reflex withdrawal to pain, right? Don't test that on your neighbor right now, okay? Please don't. The patellar tendon's all right, but so that's from, no, I'm not real tall, but that's from your toe up to your spinal cord. So let's just call that a meter. I'm going to tell you the fastest conduction, not through your pain pathway, but let's pretend through, so the sensation can go through a pathway that can travel at 150 meters per second, going for 1 meter. How long did that take? You can get your calculator out. Don't look at me. 150 meters per second, it traveled 1 meter. How long did that take? Somebody got a number? You're just waiting for somebody else to find it, aren't you? I know you have calculators on your phone. Do you know how to get that number? Does anybody want to say how you get that number? This could be a small number, isn't it? No? Oh, wait 1 more minute, I'll tell you. So quantification in science is important. I don't need an exact number, but it gives you an idea about what range of time or signaling matters, right? Right, so you give up. It's about 6, you got.
Oh, did you say it was 150 meters
per second in
so
1 meter?
Is it 0.0067?
Okay, so 6.7 milliseconds. Yeah. 6.7 milliseconds up, and of course, it's going to have to go back to the muscle. Let's pretend it goes another meter. Right? It's going to take about 12 seconds round trip, 12 milliseconds round trip, maybe 13, right? I'm round. Okay. It takes a little longer than that. So if it's 6.5, it's 6.7. So that'd be 13.4. It takes 15 to 16 milliseconds. What happened? And this is what we call a simple reflex arc. It went right to the spinal cord, fired off the motor neuron to cause that withdrawal of delay. Math didn't add up, did it? That's right in line with a synaptic delay of about 2 milliseconds. And that's what you find at almost every synapse. Every fast synapse. I'm a little slower than that. Okay? So there's a delay. So why have them? Wouldn't it be faster just to wire it up? You wire it up, get rid of that couple of milliseconds. Okay. Now, if you're a giraffe, and you have a really, really long leg, well, you're in for it, right? I mean, it's going to take a while. So it still has a delay. Why not just wire it up? Why not do what Golgi and all his followers said? It's all wired up. It's fast that way. Why have these relays? Anybody want to think about that? Why have synapses? Wouldn't it be better just to wire us up? So there's an idea there. So the idea would be that it might go off when you don't want it to. Ooh, that's a very interesting point. Did everybody hear that? If everything's wired up and all this nerve activity is going all over the place, then you could have things happening when you don't want them to happen, right? So what does that make the synapse?
Kind of a cushion.
A test, a cushion, what else? Sometimes, what's that? A regulator. Okay. Regulator's a great word. Sometimes you hear safety, right? A safety delay. We'll find that that's basically what it's doing, and that's why it's regulated up and down so much, and why some of your synapses on a given moment in time are not working, but other times they are working. So it turns out your synapses have very strong brakes on them all the time, even the chemical ones, which the vast majority are but even electrical ones. There's a break on all the time, and unless that signal is what we call salient, it has to be strong enough, it has to rise above the noise to say, okay, now I really need to move my leg away. I'm not just going to be jumping all over. Okay. Do they slow down their communication? Yes. But the trade-off is you have that regulation, you have that fine level of control. So the synapse, and what it's doing is converting those action potentials. You remember hearing that they're all or none? Have you all heard that term? Right? Shouldn't be news, right? So an action potential in almost all cells, virtually all cells, when you fire it off, okay, it has a characteristic signal, and it travels down the axon and in a smooth fashion, so it has that the fastest 150 meters per second signal velocity, okay? But it's just a bit. It's a digital signal, right? Now it comes to the synapse, and it has to have a strong enough character to release that system, but it actually does it in what we call a stochastic manner, okay? But that digital signal then has to get converted basically to an analog signal, because you're releasing a chemical, and convert it back to a digital signal. And from one nerve cell to another, that might be faithful, it might happen, but there may be other inputs that modify that. So now you're starting to see, even at a very simple level, complexity that could happen. All right. Now most nerve cells aren't driven by one synapse. There is a synapse in a squid, called the giant synapse we're going to see in a couple seconds, okay, that drives the cell. It's huge. It's the mother of all synapses. Okay. Most synapses in the brain, most nerve cells in the brain, I should say, backing up, receive many synapses. It's between about 1,000 and 10,000 synapses. Curiously, in the spinal cord, that picture I showed, you can't see the motor neurons there. But so the motor neurons, they're the last neuron that send axons out to the muscle to contract, okay? They receive about 10,000 synapses. We don't know why they receive so many, but they receive a huge number from different sources. But between 1,000 and 10,000. So if a third of them aren't working, well, maybe the other 2/3 are, right? So, but you see, you have a dynamic range in terms of how many synapses are functioning, how well they're functioning. So we talk about synaptic plasticity. We find with use, the more you use a synapse, often that will upregulate that synapse and it'll become more effective. So the next time an action potential comes, it's more likely to trigger a signal in the next cell. Okay. So that goes back a long way in terms of what Cahal did, and in the many years since, through the 20th century, there was a lot of neuroanatomy done. We know a lot about different cells, how they interact with other parts of the brain. That's not a subject that we're going to go into here. There's a huge amount of data out there on that. But a lot of it has cataloged what we know without necessarily telling us what. However, in the last 10 to 15 years, there's been a radical change in terms of how we can visualize the nervous system, and it's using this guy, which you've probably heard about. Have you all heard about the fluorescent proteins? Green fluorescent protein? Do you know what it looks like? It's an amazing structure. Have you seen this? For a long time, it wasn't clear whether there was a cofactor. It turns out that there is, and it's oxygen. So it was very hard to discover. Well, this was discovered by Osamu Shimomura, a remarkable scientist who really spent his whole career studying these fluorescent proteins. And he focused on a particular organism that was bioluminescent. This is a Coria. This was found in huge abundance in Friday Harbor in the state of Washington. This is a research site for the University of Washington. Found that the rims would glow. Okay? And they glowed kind of blue-green, depending on the conditions. Sometimes they're more blue, sometimes they're more green. Don't make a whole lot out of this picture. And long story short, there's actually two proteins in there. There's a corin, okay, which is activated, all right, basically by calcium, but that's a different story. And then the light from a corin strikes the GFP, which is shorthand for green fluorescent protein, okay, and then that gives the full spectrum of light from the rims of these jellyfish. I think they're called crystal jellyfish. And so he came up with a little tool to cut the rims off, isolate the material, the proteinaceous elements from these jellyfish, and had to use tens of thousands of them. And initially, when he tried to purify, he saw nothing. Fluorescence went away, could never find it, had no idea what was going on. Well, the clue was calcium. Didn't know about that, and apparently had been working for months and months, was getting frustrated, was going to work on something else, because he worked on other bioluminescent organisms. And one day he threw out a whole mess of this partially purified extract that he had in the sink, and he threw in, just throwing it out, some calcium chloride, went to leave, it was late in the day, turned the light off, and all of a sudden something was glowing behind him. He went back, the sink was glowing, and he discovered that calcium was the trigger for the aquarin, and that led to ultimately the discovery of GFP as a separate fraction. And then it was another scientist came along some years later and said, we could use this to visualize cells. Marty Chaffee. And he had been looking at dyes in cells and said, cells are basically translucent, light will come out of them. And he'd been saying that for years. And he suddenly realized, if you put this into cells, so this protein, the gene for it had just been discovered and sequenced. He got hold of it, was able to put it in cells and show that, in fact, the cells would light up. Well, since that time, there have been a few advances. So, Shimomura, together with Chaffee and Roger Chen, a remarkable scientist, were awarded the Nobel Prize some years ago. What Roger Chen did is he took it to the nth degree. He discovered that you can mutate the GFP, the green, and get red, or yellow, green, or blue. Actually, he's made so many different fluorescent proteins I haven't kept track. There's one called raspberry. There's tomato. It goes on. Every year there's something new. I think there's a lavender. Lavender's a weird color, but at any rate, if you take these guys, so a group at Harvard, this was Jeff Lichtman together with Joshua Saines, realized that if you had red and you had basically yellow, sort of a yellow-green, right? And then this is blue, it's seed for cyan, okay? 3 colors, you could use those 3 colors like you did in an old TV. I don't know if any of you have an old TV at your parents' house, like an old Sony. Tricolor, you can basically get all the colors you need. So they used a gene sequence. So again, they have the sequences. These are proteins. So you're using what are called genetic-based fluorescent indicators or markers or whatever, right? So have the sequence for the RFP, the YFP, the CFP, and then flank them with LOXP sites. You might remember LOXP from some molecular. Well, if you don't, it doesn't matter. These are sites that you can cleave when you use a recombinase. So you can put 3 doses of these into an embryo or into a cell. In this case, it was done embryonically, all right? And then you combine that with this recombinase, and each cell, it will randomly recombine these into different groups. So you can get a whole range of colors. And this is the first step that this is now called the rainbow system. And now it's been taken to the point where they have literally hundreds of colors as if you need them. But the thing was, each cell would have a unique recombination. And so this is really now modern day neuroanatomy. You can follow axons. This is in the optic path, optic nerve from a mouse, okay? And they split out some axons, and each one had a particular color. And now we're not talking about 5% of the cells. You can see all the cells, and you can see them in their entirety all the way up to their synapses, because that particular cell here, where's red? I have a hard time. Looks kind of purpley to me, but this one, if you look through the whole cell, okay, you can see it. Again, the membrane's translucent. The light comes out. You can see it. So you can follow from the point of origin all the way out. So now you have a code, all right? Now for that particular mouse, it's going to have a particular code. You have to work that out. But you can see all the cells. This is in the retina. You can label all the retina. And basically every cell is different from its neighbor. So this is a whole nother level of neuroanatomy. So Jeff Lichtman has taken on the task of trying to reconstruct all the synaptic interactions in the brain. It's been calculated based on the work they've done to date that it'd probably take more than 1 million years to do that, so they're going to have to come up with some other tricks. But the basic concept is, if we understand in a particular region of brain that we understand the function, okay, all that interconnectivity, then we can have a map to understanding how that then gives rise to something more sophisticated. Maybe not an emotion, but something maybe more basic in terms of that circuit. And this is being called connectomics. I know there's omics for everything, right? But so they have done this for a cubic millimeter of mouse brain, and it took them months and months, more than 6 months, to reconstruct that. Because there's already, in a cubic millimeter of brain from a mouse, not quite 100,000 synapses. Nonetheless, they will reconstruct it. So there's a very interesting approach to this now by not necessarily banking completely on connectomics using this so-called rainbow, but using basic data generated like this. So there's an idea to do this maybe in a more select fashion. So now you can label everybody. Do you want to label everybody? Well, Lichtman does, but maybe you should label certain cells that have certain function. So if you combine those two together, then you could link the function then to that interaction. So there's this neuroscientist at Princeton has come up with a way to solve this problem by basically making it an open source. And he's developed it as a game where you can go online, take a piece of data, and do a reconstruction. And his idea is, well, if you get, instead of 1 million years, you get 1 million people, maybe it'll take a year. So this really is the way of neuroanatomy in our modern times, to be able to really say, I can take that cell, I can see it where it's going, and ultimately we can say that cell does this. Okay. Now let's talk about some basic neurobiology. So all that up till now, there's some basic concepts, but we're going to revisit the synapse even now, very soon. We'll revisit some of these concepts again. But this stuff about connectomics, that's just a little bit of fun, all right? Now we got to get down to the nitty-gritty. So this will hopefully be reviewed for many of you, but it's still really quite fascinating that the nervous system basically works as a highly polarized organization, right? There is an input side and an output side. And so the dendrites are largely responsible for the input. You can get input onto the cell body, right? Or soma, it's called. So the dendrites often, they're thicker than what you see. This isn't drawn as accurately as it should be because it's, well, this is my picture. So at any rate, they're thicker, they branch, and as we'll see in a second, sometimes they have little nubs on them. You probably might have noticed that from earlier pictures. By and large, they give rise to an axon. There's always exceptions. You're going to hear them over and over again. There are cases where it looks like there may be more than one axon. But the rule, with these few exceptions, is that there's many dendrites and one axon. So there's one output. Now what's not being shown here is that this can branch like crazy, right? And it can actually go back and touch itself. It can touch other cells. We saw that collateral already, right? So now you can have direct input, you can have divergence, you can have collateral feedback, okay? And we have two flavors of input, excitatory and inhibitory. And by and large, the nervous system, as far as we understand it, operates under these two basic functional drives, okay? And by and large, excitation is what's driving circuits, okay? And the signaling in those circuits is sculpted or modified by the inhibitory action. Now, there are cases where there are spontaneous activity in nerve cells. So you cut all this off, they every now and again, they have activity and they fire a signal, they fire an action potential. okay? But most of the time, this drives it, this sculpts it. So it'll be firing along for a while, and then there'll be a big inhibition that'll happen. It'll stop it for a while, then it'll fire again. And so you can start to see already that inhibition is critical, not only for getting excitation to come out, because if this is dominant, it's not going to work, but it's all a question about when it happens. All right? So we're going to talk about patterns and timing in a second. But if this is working all the time, and this comes in off and on, then we'll get different patterns of activity. All right, we're not going to talk a lot about this yet. We're going to have a whole lecture on the action potential. Okay, so this is actually a cute little setup where you can see the flow of ions. We'll talk about that later. That's for another time. Probably, or you know a little bit about that already, I suppose. But we'll go into it so everybody will have a chance to really understand this. Okay, but the fundamental thing is we talked before about the signal flow going in that direction. So you can see that's because there is generation of this action potential or this all or none signal, this digital signal, a little blip, right? In the old days when people recorded, they had an audio monitor and you could hear the pop of each, it was set up to hear the pop of each spike. You'll hear the term action potential or spike, okay, interchangeably at times. Okay, so the key thing is we have several elements in terms of what we call neural coding, all right? This is miserable. Let me have the blackboard. All right, I'll just help. Hopefully I won't wave my hands too much. Okay, so obviously timing is everything, right? We already talked about that. If you had a signal going along, pop, 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 all of a sudden you have inhibition, all right? At that moment in time, then that would be, a burst. And often you'll see bursting. So you get a burst of action potentials. Inhibition will stop it for a period of time and then start up again. And so the timing of that then sculpts a pattern. You get a bunch of bursts. But you can have all that you can make up. You can have a condition where the frequency changes, okay? Where the frequency starts out slow and gets faster and faster and faster and then gets slower and slower. Starts out fast, gets slow, goes faster. Just make it up, it happens somewhere in the nervous system. So the frequency could change moment to moment. And so the frequency, though, is very important in terms of the signal, and that's what's shown. I think this is going to come in now. There we go. Okay. So this is something that's a bit of confusion. You think, okay, if you drive the system harder, what's going to happen? Your first thought is, well, would you get a bigger action potential? Well, no, you can't get a bigger action potential, right? They're all or none. So you can't make the output bigger. You have to make it, what happens is that you drive it more often. So what's shown here in a very graphic terms, you're going to see this, and don't be put off by this, okay? So let me start at the bottom. So here's a case where I'm not going to explain it fully, but you're recording from a nerve cell, and you can record that electrical activity, and you can record those spikes. And that's what these things are here, okay? And so these are voltage changes, and we'll talk about those details later, but there's an electrical change, all right? And it's real fast. Well, look what was related to. So here's a case where you put in a drive. We'll just say that's a drive. This is actually what we call injected current. So it's as if you put in an artificial synapse. It's an electrode, and you just drive it. And you drive it a little bit, a bit more, and then a lot. Look what happened. Initially, that wasn't enough to really drive the cell. Can you all see that? You should have that. Should be able to have that. Now you drove it, and you started to get some spikes. And look how uniform they were, because you gave a uniform signal. You're driving the voltage as if you're giving that synaptic input. You're driving that cell a little harder, and you got very, very fine, very evenly spaced frequency, right? Look what happened when you gave more. Okay? Action potentials can't get bigger, all right? This underlying bit here, this actually does mean something here, this voltage response. We'll talk about that later. That didn't really change. You've got more of an effect. So more drive translated digitally into a higher frequency. So frequency is a code. The higher the frequency, it's telling you that circuit is being more strongly stimulated. Okay? When it happens is the timing. And that's all over so the signal started here and ended there. And you can, do short ones, long ones, all different patterns all over the place. Now there are cases, I will tell you, where you do this and you'll get increased frequency, but then you'll start getting bursting. So this will burst for a while, it'll stop. It'll burst for a while, it'll stop, even though that's what you put in. And that's because of the underlying machinery that's there. So each nerve cell has a unique set of ion channels that translate into this signal. But back at the level of the dendrites in the cell body, they differ. So in some cases, they have little feedbacks. So they'll excite for a while. This is without that inhibitory input, okay? Just exciting, it'll go for a while, and then at the cell membrane, it will stop. Then it will start again. So there can be inherent bursting, there can be bursting that is in fact sculpted by the inhibitory input. And the last thing, so obviously this is in a box, so that must be important, right? So there are a lot of things that I'm presenting today that are important, but we're going to get back to the synapse and the axon and the axial potential. But we're really never going to talk much again about signaling in the nervous system, this neural coding, okay? And the last one we've been alluding to all along, right, are patterns, right? And there's all kinds of patterns. So that pattern then could translate into something, whether it's an emotion or just some kind of reaction. So timing, when it happens, frequency tells you a lot about the nature of the signal, particularly in that pathway, the strength of the signal, all right? And then the pattern, without explaining it, it's different all over the place, but that inherent in that pattern are the subtleties of signaling. And this is all really at the level of the axon, just signaling down the axon. We're ignoring the synapse. Now the synapse's job, even though I told you it doesn't work all the time, and it's highly dynamic, it's still to convey the message. It just does it in a variable fashion. So ultimately, these really are the signals, right, that ultimately are going to drive the system. Okay, so in the last few minutes, actually I'm going to stop. Are there any questions? Because, again, box looks like it's important, huh? Looks like it could be a question on the test. Is there a question up here? He's just stretching back there. Yeah.
What's the exam format? Is it like multiple choice?
You want to know about the exam already? It's a combo. So we used to do actually multiple choice and then short answer essay. We can't handle that. Okay. So we'll have short answer. Now, I still have a little bit more to go, by the way. We're not done. I was just stopping for a second. Just give me a few minutes.
Wait, so
You can start moving if you want. Just go real slow. Yeah, I'm going to wrap up. But so we'll have the multiple. It's going to be mainly multiple choice because you're a big group. But then we have short answer that are going to be like match or fill in or selection. Okay. So no essays. Your essay is going to be that topics thing. But we'll go over that again. So let me just show you a few cool things and we'll be done. One is this actual picture of a synapse. So there are certain components of the synapse that are essential. This is again all on the PowerPoint. Things like vesicles that carry the neurotransmitter that transmit that electrical signal from one cell to another through a chemical spot. So this gap here is really, really small. It's like 50 angstroms. Okay? So it's really tight. There's an electron micrograph picture of that. Okay. Now, what we didn't show you before, is that for excitatory synapses in particular, all right, don't see with inhibitory ones, there's a little bump that comes off the dendrite, and that's shown here. See, this is actually a dendrite, a stalk, and this is called a spine, okay? So often, this is called a presynaptic element, almost always has mitochondria because you need ATP for generating this communication, okay? And then there's receptors on the other side for the signal, all right? But often there's a bump there, and they actually have functional elements that are involved with it, okay? Now, as you already know, the key signal for this communication, and I'll come back to this, but this is no surprise, it's calcium. Calcium's everywhere, right? But those calcium channels are oriented right in that synaptic cleft, right at the site where you get the release. We're going to come back to that. But the point here is all this is highly orchestrated. Often you look in the text and you see this, and you see vesicles of different sizes. Well, they're actually pretty uniform. They vary to some degree, but they're much more uniform with this, and they dock at particular sites, and how they communicate is highly orchestrated. Okay? I'm going to skip this because of time. This is just showing a voltage relationship between the input cell, the presynaptic cell, and the output cell, and the switch ion synapse, and the timing. So you can look at that later. But I just want to show you this, and we'll quit. So this really has been the most exciting thing in neuroscience in terms of being able to do neurobiology at the level of circuits that has come along in a long time. I mean, really in the last 3 to 5 years. It goes back about 10 years. And that's using what's called optogenetics. And this would be something that we're not going to cover much later, but this could be a great topic for any of you who want to delve into. So instead of recording with an electrode, okay, where you put in current and you can get action potentials, we can now see them. So you use again, a genetic sequence, a gene. In this case, up there, it's a sequence that has the voltage sensor in a channel. We'll come back to what that is, but channels respond to voltage, but the molecules move. So they move, they basically bump the fluorescent proteins so you get a burst of fluorescence. So what's shown here is that you can visualize an action potential. You can see it, rather than have to have a big amplifier and a big recording system to get it, okay? And here's a case where you can get these action potentials, in this case, 23 separate cells at the same time. Normally you put an electrode in, you record from that cell. Some people made electrodes where they have like 8 or 10 of them, try it, and they don't work so well. In this case, you can visualize them all at once. So it's extremely powerful means now, can interface with those connectomics, where you now functionally understand that circuitry. Now you can look at the action potentials in a whole circuit. Okay, and I'm not going to go through the explanation for this, but not only can you use this to detect the voltage, you can use this. You can use the same approach. Okay, that is an optogenetic approach in the opposite fashion, where you now use light to excite the cell or inhibit the cell. So you can drive action potentials or not. So not only can you see the action potentials, you can visualize that functional circuit. You can now manipulate it. You can take these 3 cells over here and fire them, take these 3 cells over here and inhibit them. This is a demonstration of that. And the last thing I want to show you is what we can really visualize down, not just at the cellular level, but the molecular level. I know this looks awfully complicated here to the left. Don't worry about that, but what this allows you to do in an intact animal, you can visualize the dendrite and these spines, and these are days, 70 days, 106 days, 126 days, 166 days. For days on end, you can visualize that cell and see how it changes, and particularly these spines. And it turns out your spines are changing all the time. And the last thing I'm going to show you here before we quit is the fact that we can use this visualization now to not only see these spines, so there's a dendrite with the spines, we can look inside the spines and we can see using super resolution microscopy individual molecules. And last year the Nobel Prize in Chemistry was awarded to 3 individuals that pioneered this technique, where now we can visualize all the way down to the molecular level what's going on inside of a part of the synapse. Now we can go all the way the other way, there's MRI and PET scanning, but we're not going to really cover that here. So somehow, maybe one of you someday could figure out how to pair, all the way down to the molecular level, understanding the synapse to whole brain behavior. Okay, we're done.